Alright, ladies, so this video is going to go ahead and get us started on the last part of genetics, which is chapters 14 and 15. And this is going to cover some select material that appears in these two chapters. So it won't include everything that's in there, but what I think are the most important parts. And this is going to cover human heredity. So now we talked about Mendel, we talked about what DNA is and how you use it. Now we're going to look at some more details in terms of the human genome that you have. So our essential questions, first big one is, how do we use genetics to study human inheritance in particular? And so we're going to look at this from sort of the same angle of inheritance, but now we're going to focus on humans. And along with that, we're going to do something that I think you'll find interesting, which is to look at a lot of different genetic disorders as well. The second part of this, which is more chapter 15, is to look at how and why scientists or doctors manipulate the DNA in living cells. So how do we work with specific genes, and what is genetic engineering, and how is this applied to humans today? And then what is also kind of interesting is, how does this lead us toward ethical debates in terms of what's right and what's wrong in a lot of scientific research. Okay, so this first statement here is very true, and that is that one of the most mysterious creatures in the world is one called Homo sapiens. And I'm hoping that you know that that's you, right? Humans are not really well understood in terms of how our bodies work, how our brains work, just a lot of it, and it's because humans are very complex animals, and so scientists still have a whole lot to learn about us. Now, in recent years, and I know you guys are young, but really just in the past 30 years, scientists have begun to understand humans a lot better by looking at their genetics. And we say that what we look at is the human genome, where a genome is all of your hereditary information. So all the chromosomes you have, all the genes you have, make up your genome. Now, one of the best ways that scientists have found to study the actual chromosomes that you have is called a karyotype. So don't confuse this with genotype and phenotype from before. This is actually something different, and a karyotype is actually a picture of your chromosomes. So in a karyotype, which we'll see here on the next slide, scientists are actually going to take a picture of a cell that's in mitosis, cut the chromosomes out of that picture, and then arrange them with their homologous pairs. So again, remember that you have a set of chromosomes that originated from your mom, a set that originated from your dad, and all those pair up. So you have one that's just like one that's very alike from both your mom and your dad, and that's what made up a homologous pair, as we saw in meiosis. Now, the first question is, why is it a cell in mitosis? And in particular, scientists seek out cells that are in metaphase. If you think about it, it's because the chromosomes are so easy to find. Right? Remember what metaphase looked like? Big cell, chromosomes lined up right in the middle. They're there for the taking, right? So if we find a cell that's in mitosis when the DNA is condensed, so we'll kind of write that here, right? DNA is condensed during mitosis, easier to see. And then we find one in metaphase where it's all lined up. It's going to be really easy to get the chromosomes out. Now, the classic way to do this for scientists was really to take a picture through a microscope, blow it up bigger, cut the chromosomes out with scissors, and then glue them back with their pairs. Today, as you might guess, this is done digitally, so you use a microscope still, but then you use a computer to help you isolate the chromosomes and separate them and things. Now, when we look at one of these karyotypes, you're going to notice that there are 23 pairs of chromosomes, as we've already learned, right? You have 46 total chromosomes that appear in pairs, so 23 pairs. The majority of these chromosome pairs are numbered, and that's how we label them, right? So you have chromosome 1, chromosome 2, chromosome 3, and so on. And they're numbered from 1 to 22 based on size. So the longest chromosomes are like chromosomes 1, chromosomes 2, chromosome 3, and the shortest ones are 20, 21, 22. Now, these 22 numbered groups, for a total of 44 chromosomes, are called autosomes, or autosomal. That leaves you just one more pair, your 23rd pair, and that is your sex chromosomes. And maybe you already knew this or maybe not, but the sex chromosomes are not numbered. They have letter designations. You can have either an X chromosome or a Y chromosome in your sex pair, and then you have two of those. So if you are XX, you are female, and if you are XY, you are male. So at that pairing, girls have 2 copies of the X chromosome. At that pairing, males have 1 X and 1 Y. So again, you've got 22 numbered pairs called autosomal pairs, and then you have 1 sex chromosome pair. Now, I couldn't come up with a good picture for this slide, so you got Usher, because he's sexy, or something like that. Okay, so here are what karyotypes look like. Here on the left is a normal female karyotype. You have all 23 pairs of chromosomes lined up, and they're numbered by size. So smaller numbers, bigger chromosomes, larger numbers, smaller chromosomes. And then down here in the bottom right corner, you'll find your sex chromosomes. So for the female, like I said a moment ago, that's going to be XX. So notice this one has 2 X chromosomes circled here, so that's a female. Here's a little bit smaller picture, but here's what the male karyotype would look like, or a normal male. Again, 22 numbered pairs, then the sex chromosomes, and in this case you've got XY, so that's male. And notice this is a weird chromosome pair. The XY pair in males is the only homologous pair that's not really homologous, right? This X chromosome is way bigger than the Y chromosome, but still that's the pair you have. Now the other weird things you see in this picture where some of them are like bent, like you see right up here, and things, that's just natural, right? Remember, this is a picture of the chromosomes, so they might not just be a nice little perfect shape inside the cell. Now these are also non-duplicated. Right, we've removed that duplication piece. So instead of looking like X's, each chromosome just kind of looks like half of an X, right? And then you pair those together. Now, the way we then classify humans is we write their total number of chromosomes followed by their sex complement, so whatever sex chromosomes they have. So a normal female is written like this, right? 46XX. Sometimes we don't have that comma either. We just say 46XX. And a normal male is written as 46XY. Now, if there's something wrong where you have an extra chromosome or you're missing a chromosome, then that'll change this number or it'll affect your sex complement, and so that is shown in these classifications. Now, another way that scientists study human genetics is through a pedigree chart, which many of you have probably seen in previous classes before. If this wasn't biology, we'd probably call these charts family trees, but the specific biological way that we draw them in a very standard form is called a pedigree. Now, we talked about Mendel a lot already, but when we talked about Mendel, we were really applying his ideas to the plants he studied or little animals, that kind of thing. But really, Mendel's inheritance does apply to humans as well, just for not very many traits. Right? As we talked about, most human traits are polygenic, right? Affected by many different genes. But a few traits are actually controlled by one gene. And I think I mentioned these kind of offhandedly back in class before. Again, if you give yourself a thumbs up, right? If your thumb points pretty much straight up in the air, then you have a quote-unquote normal thumb. If your thumb bends so that you point almost backwards up your arm, then you have a hitchhiker's thumb. And this is a trait in humans that's believed to be controlled by just one gene, so it follows Mendel's laws. Same thing with the presence of dimples in your cheeks. Same thing with whether your earlobes are loose from your head or connected to the side of your head. Same thing with widow's peak. If your hairline in the front comes to a sharpened peak, that's just controlled by one gene versus not having that peak. and there's several others as well. So if you have a trait, or in some cases a disease, that's caused by one gene, it follows Mendel's rules, and you can draw a pedigree chart for it. So here's a basic pedigree. This little guide appears in your textbook, showing male and female who are married or mating, and then their offspring. And so this, you can kind of read in your notes those pieces, and the next slide walks you through these pieces as well. Now, the people who use a pedigree are people called genetic counselors. And genetic counselors might be physicians, they might be doctors, or they might just be real geneticists, right? Have an advanced degree in genetics, and they study genetics in humans, and then they serve as counselors. They work with people, couples, things like that, who have maybe a genetic issue themselves, or have some questions about genetic diseases that might run in their families, and things like that. And a genetic counselor might construct a pedigree for your family and try to make some predictions based on that. So here's sort of your key to how to interpret a pedigree. Males are squares, females are circles. If you shade in the square or circle, that means that that individual has the trait that you're talking about. If you don't shade that individual in, then it means they don't have the trait that you're talking about. Something else you can see, I'll draw this down here on the side, is sometimes you might see a shape that's halfway shaded in. If you see that, that means the individual is a carrier, which is kind of another way of saying they're heterozygous, right? So maybe they have this kind of genotype, and by half shading it in, it tells you that they're actually kind of half and half. Right? So you don't always see that, but sometimes you see that. Now, some of the other things you might notice, you might see a diamond, and that means person whose sex is not known. That sounds really weird. It doesn't mean that you look at the person and you can't tell if they're a boy or a girl, which I know happens if you go to the mall. But what it means is that you put together this pedigree maybe by asking a family about their ancestors and the people that came before them. You might be trying to put together a pedigree, and you're talking to a person, and you're like, okay, so tell me about your great-grandparents' brothers and sisters. And their response might be, well, I know that my great-grandpa had 2 siblings, but I actually don't know if they were brothers or sisters. Well, you can include them in the pedigree, but you would write them as diamonds, since you don't know if they're males or females. Now, a single line directly connecting a male and female indicates that they are married, although I really don't like that. It indicates that they're mating, right? I know we're at a Catholic school. We want to tell you that you should be married if you're mating, but in biology it doesn't work that way, right? Because we could be talking about any kind of pairing here. So a line between the two means they've mated. 2 lines between the two means they have a consanguineous marriage. Take a moment and try to guess what that means. Yeah, if you said you're married to your relative, you are correct. So lots of consanguineous marriage here in Kentucky, right? Go Cats. I'm teasing, but if you are mating with someone you're closely related to, maybe a first cousin, maybe a sibling, maybe an aunt or uncle. I know that's creepy and nasty, but it does happen, and so then you would draw these double lines between the two. Now, then, if you're talking about offspring over here on the right, a vertical line, like you see here, indicates that that's their child, right? A vertical line that's then bracketed, like you see in this one, shows multiple children for a family. And then you also have twins. I'm really not worried about this. I won't ask you any questions specifically about twins. But this top one is the way you would write non-identical twins, and this is how you draw identical. But again, I won't ask you any questions about that. Now, you would draw a pedigree to sort of follow a trait or follow a disease through a family, and then try to figure out what the genotypes of those family members are. So basically, a pedigree you draw by gathering phenotype data, right? Do they have a certain trait? and then you use that information to try to figure out what their genotypes are. So you can figure out if someone is homozygous recessive, right, or homozygous dominant, or heterozygous. Now you see that this genetic stuff never goes away. Now, pedigrees also allow you to determine whether a trait is dominant or recessive, like we've seen before, or sort of our new thing here, whether a trait is autosomal, meaning it's transferred on those numbered chromosomes, or sex-linked, meaning it's transferred on the X chromosome. Now, as we stated before, a lot of your traits are polygenic traits. They're controlled by lots of different genes. So those we can't really put into a pedigree, just like we couldn't really put one of those into a Punnett square. And also don't forget that the environment plays a role in your traits. So we might not always have a perfect pedigree because it's not all about your genes. A lot of it has to do with your traits as well, or your environment as well. Now, ultimately, all we do with a pedigree is it allows us to sort of observe a whole bunch of Punnett squares. Now, I'm not going to make you draw Punnett squares at all during this chapter, but there are times where you might want to, and if so, go for it, because that'll also help you kind of make sense of what you see in a pedigree. Now, your homework along with this is going to be to work on a worksheet that will guide you through some pedigrees. But I want to give you some background when you look at these to try to figure out what's going on. This slide is sort of the three major questions you should ask yourself when you're trying to figure out or interpret a pedigree. And then I've got one more slide here where I've just got a very generic pedigree, and I'm going to kind of walk you through that one. So the first question that you're usually going to be asked is the pedigree you're looking at dominant or recessive? And here's what you want to look for. Okay? If that pedigree is dominant, meaning that it is showing you a dominant trait, so the shaded people are have a dominant trait, you're going to notice a couple different things. A dominant pedigree is going to include half or more shaded. So when you just glance at a pedigree, if half or more, so in other words, if a majority of the people are shaded in, it's probably a dominant pedigree. The second thing you can look for is in a dominant pedigree, you will see shading in every generation. So the grandparents, the parents, the kids, the grandkids, each generation you will see some shading. If that's true, it's probably dominant. Now, your other option is for it to be a recessive pedigree. Now, if it's recessive, usually you will see less shading. So in a recessive pedigree, the ones who are colored in are the people who have the trait. Recessive is usually a little less common, so if you see a pedigree where there's very few people actually shaded in, it's probably recessive. The second thing you can look for, which is just as valuable, is does it skip generations? If shading skips generations, then that's a really good sign that it is a recessive pedigree. And we'll kind of walk through this on the next slide again. Now, once you've established number one, you can move on to number two. Who is homozygous recessive? Now, this will definitely, I think, make sense when we look at an actual pedigree here in a moment. It'll make sense better. But in a dominant pedigree, the shaded in people are dominant, right? So in a dominant pedigree, the shaded in people are going to be, say, big T, or big T, little t, which means that the non-shaded people are homozygous recessive. So if the pedigree is dominant, then the homozygous recessive people, or little t's, are going to be your non-shaded people. But if you're looking at a recessive pedigree, in this case, the shaded in ones are going to be the little t people. Right? So if it's a recessive pedigree, then the shaded people are the ones who are little t, little t. Now, once you've established those two things, then you want to ask yourself, what do you know about everyone else? And you'll always know one of their alleles, right? You might not know if they are big T or big T little t every time, but you at least probably know that they have a big T or they don't have a big T. Alright, so here's just a generic pedigree. We'll say that this is for widow's peak. Okay, so if you have a person who's shaded in, they have a widow's peak, so they have vampire hair, right? It just comes to a peak in the front. That doesn't mean they're a vampire, right? A lot of you guys, ponytail here, also might have a widow's peak in the front. But this means that the people who have a widow's peak are the ones who are shaded in. And so the people who are not shaded in do not have a widow's peak. Now, I would ask you a particular couple questions about this pedigree, but all of them are going to take us to basically those three things that I had on the last slide. So, first question, is this dominant or recessive? So, let's see here. Whoa, that's not what I wanted to do. Get rid of that. Alright, so number one, dominant or recessive. Now, to figure that out, we can just look at the pedigree. Again, do I see half or more of my people shaded in here? So, are a lot of the people shaded in? Looks like, yeah, half of them, right? Second thing, does it appear in every generation? Yeah, right? You have shading here in the first generation, you have shading here in the second generation, and you have shading here in the third generation. So, if those two things are true, then it's probably dominant. So go ahead and circle that. Right? Now, if there were very few people shaded in, say maybe just one person or two people, and if we had skipped a generation, so maybe no one in that middle row was shaded in, then it's probably more likely to be recessive. Now, on the last slide, we said if it's a dominant pedigree, then those who are shaded in, right, so if it's dominant, shaded people are going to equal big T, or big T, little t. And if that's true, then unshaded people are going to be little t. So, I can add in some of these genotypes right now. Right? Anybody who is not shaded in is homozygous recessive, little t. So my question might be, what is the genotype of a certain individual? And now you're halfway there. Now also the way that we usually do this, and you'll see this on your worksheet, is each generation is labeled with Roman numerals. So you'd have generation 1, generation 2, and generation 3. And then each person is numbered with regular numbers, Arabic numerals, whatever we call those. So let's say my question would be, what is the genotype of person 1, 2? Right? And so you'd say their genotype is little t, because that's talking about this person. Now, the third question from the last slide was, what do I know about everyone else? So if everything else so far is true, then all these shaded people are either big T, or big T, little t, which means all the shaded people have at least a big T. Now here's where it gets really fun if you're a puzzle kind of person. If you know people's kids or people's parents, you can figure out these genotypes further. So for instance, let's look at this first family up here on the upper left. if mom is two little t's and dad has at least one big T, and then we look at their kids and they have kids who are little t, that means mom and dad both had to give a little t. So we know that dad is big T, little t. And for those kids, we know they're also big T, little t because mom had to have given them a little t. Now, same thing for the family on the upper right. If this mom right here can have a kid who's homozygous recessive, she must be big T, little t. And if this girl right here has a dad who's little t, then she must be big T, little t. Now, the catch comes with this person. Because all we know is that her parents are both heterozygotes, she might be big T, right? Or she might be big T, little t. So for this person, we'd write big T question mark. Because we don't know what the rest of her genotype is. But we do know that she has that big T, which got her shaded in. Now, I know that's a lot to digest. We'll do a couple different assignments and some different things in class to look at these. So hopefully you can work through this first pedigree worksheet. It'll kind of walk you through a little bit at a time, and you can start to make sense of this. So thanks for watching.